Good morning, friends. Good morning, students. In today's class, we'll discuss replication, right? So this lecture is on replication in prokaryotes. In the next video, we'll discuss replication in eukaryotes, right? So first of all, I will start with the definition. So what is replication? Replication is synthesis of new DNA, right, from parent DNA, right? Or we can say that it is formation of copies of DNA from the parent DNA, right? Now we'll start with the salient features of replication. Now the first salient feature is that the replication is semi-conservative, right? Semi-conservative means half of the original DNA is conserved, right? So during replication, when the parent DNA forms daughter DNAs, the one half of the original DNA is conserved, right? So this is normal DNA strand in which the one strand is blue in color and other is black in color, right? So when replication proceeds and its daughter copies are formed, so one strand is blue, so other strand will be formed, and in similar manner the one strand is black and there will be formation of one new strand in this, right? So this parent DNA is there and these are the two daughter DNAs, right? And since one strand is blue in color and other is black in color, so in this one of the blue strand is there, it is semi-conservative, and one strand is black, so half of the original DNA is conserved, right? So this was proved by the scientist Michelson and Stahl experiment, right? Now we'll start with the second feature, that is initiation of replication. Now the initiation of replication, right, starts at the origin of replication, right? And in prokaryotes and eukaryotes there is major difference that in prokaryotes there is a single origin of replication, and in eukaryotes there are multiple origins of replication, right? And there is one salient feature is that this site where original replication take place, it is highly rich in AT rich base pairs, right? So original replication has high quantity of AT rich base pairs, and there is specific protein called DNA A protein, right, with 20 to 50 monomers. It's attached, and that starts the formation of replication bubble, right? Now coming on to the replication bubble, in suppose we unwind the DNA and there is formation of replication bubbles in the replication site, right? Now these are two complementary DNA strands, one is into 5' to 3' direction, other is in 3' to 5' direction. When these two complementary strands they separate at the origin of replication, that results in formation of replication bubble, right? But in prokaryotes there is single origin of replication, there is not formation of multiple bubbles. But in case of eukaryotes here, there are multiple replication bubbles are there, right? Now coming on to the RNA primer, right? For the synthesis of new DNA, we need a small fragment of RNA with 5 to 50 nucleotides, right? For the synthesis of new DNA, right? Now in leading strand, there is single RNA primer is required, right? And there is continuous synthesis there. But in lagging strand, there is multiple RNA primers are required, and there should be continuous synthesis of RNA primer. Now coming on to the next feature, that is it is semi-discontinuous and bidirectional. Semi-discontinuous means that it can continue with some breaks. So in case of leading stand, it is continuous, but in case of lagging stand, it takes place in breaks, so it is semi-discontinuous, right? And coming on to the bidirectional, right? DNA synthesis take place in both directions, right? How can you learn it? For example, this is a highway and we have a divider inside, right? On the highway, one side is this side, another side is this side, right? So bidirectional means when you are going from Amritsar to New Delhi, one road is going upside, another is coming downside. So in this way, you can compare 5 prime to 3 prime direction synthesis, and on the other side 3 prime to 5 prime, but it is anti-parallel. So you can imagine it, remember it by this way, right? Now I will start with the role of DNA helicases, right? Helicase means to unwind, right? To form replication bubble, there is formation of a replication fork. So there is normal DNA, which is 5 prime to 3 prime and 3 prime to 5 prime, right? So here, when we form replication bubble, a replication fork, there is attachment of this enzyme DNA helicase, right? The helicase will keep on opening, it will attach here and it will keep on opening the, or keep it act as a zip opener. It opens the two strands of DNA. As it will move further onward, the DNA will start to keep unwinding itself, so it will keep opening and this will keep moving forward in the direction and DNA will start to open, right? And when the two strands they open, uh, on the outer side of the DNA there are attached some, these are known as single stranded DNA binding proteins, right? They attach themselves on the outer side of this bubble to that prevents the single stranded DNA from attack of the nucleases and keep them stable. So these blue ones are this SSB or single stranded DNA binding proteins, and this red one is helicase that acts as zip opener and is opening the DNA. Now we'll start with proper application. Now to remember proper application, I will give you one mnemonic, that is you will always see template in the direction 3 prime to 5 prime direction, right? T for template and T for 3. So template is that is in 3 prime to 5 prime direction, and DNA synthesis as resembles 5, so DNA synthesis will occur in 5 prime to 3 prime direction, right? So this is the basic diagram of replication. There is one 3 prime to 5 prime strand and the other is 5 prime to 3 prime strand. So these blue are anti-parallel, 3 prime to 5 prime and 5 prime to 3 prime, right? So when we will start unwinding the strands, the first comes the role of DNA helicase, right? So DNA helicase act as zip opener and it opens the two strands and the zip is opening and it is moving forward in this direction, right? So when DNA helicase unwinds the two strands of DNA, the two single stranded strands are stabilized by these SSB or single stranded DNA binding proteins. These are shown in the as blue ones. So now the single strand is stable, right? Now comes the role of RNA primer. So this one is the black one is RNA primer, or RNA primer their synthesis of new DNA, right? And we remember that synthesis takes place in 5 prime to 3 prime direction and anti-parallel to the parent strand, right? So this is 3 prime to 5 prime, and this black one, since helicase unwinds and the single stranded strands are stabilized by SSB, and this black one is RNA primer or RNA primer. There is synthesis of new DNA in the 5 prime to 3 prime direction. The new nucleotides are added towards the 3 prime side, right? And this blue one and black one is DNA polymerase 3. So it will keep adding the new nucleotides at the 3 prime side, right? But on this strand is leading strand, and the downward one, this one is lagging, right? The difference is that on leading strand there is continuous synthesis, and on lagging strand there is semi discontinuous synthesis, right? The problem is that in this direction we are synthesizing regular, and in this discontinuously. Why? Because DNA polymerase 3 can add in the 3 prime direction, right? So in this direction, as the helicase is unwinding, a zip is opening, it will keep on moving, and on 3 prime end we are continuously adding new nucleotides, and DNA polymerase is doing its role. But in our human body we don't have DNA polymerase that can add nucleotides to 5 prime side, right? Our body doesn't have. So in this case the zip is opening in any direction, but we cannot synthesize in that direction because this is complementary. 5 prime is on this side and 3 prime on this side, right? So it will accommodate by synthesizing them in small fragments in 5 prime to 3 prime, 5 prime to 3 prime, and it is anti-parallel, right? So in case of leading strand there is synthesis so in the direction 5 prime to 3 prime direction towards the replication fork, and in lagging strand there is synthesis again in 5 prime to 3 but away from the replication fork, because the body doesn't have DNA polymerase 5 that can add to the 5 prime side, so the body can add only to the 3 prime side, right? So leading strand is there, lagging strand is there, helicase unwind the DNA, and SSB are stabilizing the single stranded DNA. RNA primer on which synthesis is taking place, and synthesis is taking place in 5 prime to 3 prime direction by DNA polymerase 3. So strand is elongating and the helicase is unwinding, so strand is being synthesized in this direction. And in case of lagging strand, synthesis take place in 5 prime to 3 away from the replication fork, because you cannot add nucleotides on the 5 prime side because body doesn't have that can add to the 5 prime side. So it accommodates by synthesizing in the 5 prime 3 in away from the replication fork. So it is synthesizing in the form of small fragments. These fragments are known as Okazaki fragments, right? So when there is synthesis of by DNA polymerase 3, there is long strand is being synthesizing, and after that comes the role of DNA polymerase 1, right? So when you will remove RNA primer at that point, you will add, for example, this is DNA, right? And this is DNA polymerase 3, which is synthesizing in the direction 5 prime to 3 prime, right? So here we have black one RNA primer on which we are synthesizing the DNA, right? So when this RNA primer is removed, when we'll add DNA polymerase 1. When DNA polymerase comes into action, the RNA primer gets removed, right? So this is 5 prime to 3 prime direction, and from here RNA primer is removed by DNA polymerase 1, right? So in case of Okazaki fragments, we have small fragments. So there is one small fragments, and these are attached by DNA ligase, right? So DNA ligase ligates the strand, and there is formation of, so suppose there is one blue colored strand here, the blue one is DNA ligase, it will ligate them, and there is formation of continuous strand, that is the DNA, right? Now since this helicase is unwinding the DNA, there is formation of supercoils at the back, right? And the problem of supercoils is overcome by DNA topoisomerases, right? There are two types, DNA topoisomerase 1 and type 2, right? In case of topoisomerase 1, it targets the single stranded DNA, and then it ligates it, overcome the problem of supercoils, and then it ligates them. But in case of type 2 topoisomerase, it can act on both the strands, right? And it also has ligase activity, right? And it is also known as DNA gyrase, right? DNA gyrase. And what now coming on the uses of topoisomerases, they are used in treatment of cancer because we use several drugs that target this topoisomerases to control the uncontrolled multiplication of cells. So that finishes with our application of prokaryotes. So keep watching, keep studying. God bless you, and do like and subscribe my channel and share with your other friends as well. Thank you. God bless you.